The previous lecture concerned the mitotic clock that drives the rapid, repetitive cell cycle appropriate to the early embryo. And now I want to develop a sketch of a complete cell cycle appropriate perhaps to somatic cells. Fix up my lines just a bit there. The embryonic cell cycle consists of just M phase, where M is for mitosis, and S phase, where S is for synthesis, specifically synthesis of DNA. S phase is when DNA replication happens. As the embryonic cell cycle eventually slows, gap phases are added to this, or growth phases. G for growth. There's G1 here, and G2. There might be different durations of these phases depending on the context of the cells. There are also two major off-ramps here. One is a parking lot, in effect, G0, no growth, a sort of a waiting state. Most of our cells, most of the time, are probably in G0. This one here that I've diagrammed, this off-ramp is for differentiation. You could think of that as D phase of the cell cycle if you want. For many cells, differentiation means a terminal exit from repeated cycles of growth and division. Not necessarily for something like this color cell here, which could perhaps return to division. But for, say, differentiation of a cell into a neuron, that's probably exiting the cell cycle more or less for good. Now, the stages of mitosis are, we can map them roughly on here like this: prophase, metaphase, anaphase, and telophase in turn. Again, the embryonic cell cycle bypasses these growth phases. There's no growth in the embryo until the embryo manages to open a mouth, becomes a larva, and starts to take on mass. It's all just subdivision of what mother managed to stick into the egg, right? So just as the embryonic cycle skips some phases of the cell cycle, many cells in animal development undergo endoreplication. That is, they might skip M phase, conduct repeated rounds of DNA synthesis without actually going through chromosome segregation and cytokinesis. Most of the cells of insects and other arthropods conduct repeated rounds of DNA replication like that. They endoreplicate to increase their biosynthetic capacity without actually going through cell division. Incidentally, meiosis, although it's just two loops through this, consists of something like mitosis, but skipping the rest. On this cycle, now I want to map on checkpoints. We already talked about an important checkpoint that gates passage between metaphase and anaphase. That's the spindle assembly checkpoint. The mitotic cell assesses, and at least for small cells, this is demonstrably the case. The mitotic cell assesses by listening for signals from each individual kinetochore whether the spindle has assembled correctly, that it's bipolar, and that it's caught every chromosome pair, every pair of chromatids, excuse me. The so-called SAC is as it happens, based upon the inhibition of the anaphase promoting complex, keeping MPF activity high until all of the conditions are met, the conditions principally being the alignment of chromosomes at the metaphase plate. Other checkpoints include several that gate passage through G2 and into M phase. There's a checkpoint, for example, a couple of checkpoints that assess DNA damage and will hold up the cell cycle if damaged DNA is detected. The SAC is based on, like I said, APC inhibition. Based on the previous lecture, you might guess correctly that entry into a checkpoint that gates entry into mitosis might be based on inhibition of CDC25. Other important checkpoints gate entry into S phase. There are mechanisms that check the integrity of DNA, nutrient conditions, whether the cell basically has enough mass to proceed with division, various stress checkpoints, etc. Many of these are based on inhibitors of the CDKs, controlled by a group of proteins that I'll discuss in a minute. CDK, controlled by proteins like P53 and the retinoblastoma protein. Cyclin B CDK1, that is MPF, is principally concerned with driving the cell through M phase. Other phases and transitions in the cell cycle are in turn governed by other CDKs and their associated cyclin subunits. These include the cyclin E or cyclin A complex with CDK2, that's supposed to look like a hammerhead shark. These drive the cell through S phase. And to drive the cell from G1 into S phase, we have the cyclin D CDK4 complex. And there are others. These are the principally relevant ones for this story. In a similar way to the previous lecture, I'm going to chart out an abbreviated version of the circuit that cyclin E is involved with. Cyclin E is transcribed in response to a transcription factor called E2F. So E2F promotes transcription of cyclin E. Now E2F can be phosphorylated in response to cyclin E CDK2, and that phosphorylated E2F is rapidly degraded as it happened. E2F might also form a complex with the retinoblastoma protein, so-called because it was discovered as mutations were in this protein are causative for the cancer retinoblastoma. It can form a complex with the retinoblastoma protein, in which complex E2F behaves not as an activator of cyclin E transcription, but as an inhibitor. RB can be phosphorylated. Phosphorylation of the retinoblastoma protein inhibits it from functioning this way. It can be phosphorylated by a number of different things, including cyclin E CDK2. It can also be phosphorylated by cyclin D CDK4. Now, I want to say this is an out-of-date and incomplete picture. This is how we thought it was working some 12 or 15 years ago, basically. And subsequent discoveries added a major wrinkle to this that accounted for experimental data that this could not. But what this basically amounts to is the following. Cyclin E CDK2 is inhibiting the function of retinoblastoma, and the RB protein in turn is inhibiting, in effect, cyclin E. That is, we have a double negative either or sort of condition between these two factors. Whereas E2F promotes cyclin E production, but cyclin E in turn promotes the degradation of E2F, that is, inhibits E2F. So we have, in effect, cyclin E inhibiting E2F availability in this kind of single negative feedback loop that we know is prone to generate oscillations. Again, we have a couple of simple intertwined feedback loops. This one tends to generate oscillations, and twining it with this feedback loop tends to increase the amplitude of those oscillations. And the key role for cyclin D here, drawn rather illegibly, excuse me, a key role for cyclin D is to kick this circuit out of the inhibited state, the retinoblastoma, the RB inhibited state, and therefore trigger cyclin E expression to get cells into S phase. I'm going to stick to the middle of the board here to keep this diagram in view while I highlight another major function of the retinoblastoma protein. It is one of the major restraints on cell proliferation. Let's imagine somewhere in the genome the cyclin E transcription unit, the cyclin E gene. Now, E2F normally functions as an activator of cyclin E transcription, but the retinoblastoma protein hoods that signal in effect, preventing E2F from activating cyclin E. So that's the retinoblastoma protein there. This is E2F. And we get no transcription this way when the complex is bound. Now, imagine a receptor on the cell surface awaiting a signal coming from who knows where to trigger cell proliferation. So let's say this receptor binds its ligand, some sort of growth factor, that will initiate an intracellular signaling cascade of some sort, probably going through RAS, small GTPase, and ending up with the phosphorylation of RB. So RB is going to release from E2F. Phosphorylated RB does not bind to E2F. The unmasking of E2F signals for the recruitment of RNA polymerase, which will then initiate the production of cyclin E and stimulate entry into S phase. Hence, when RB is absent, it's as if the cell has received a signal from some sort of what we call a mitogen, that is something that stimulates mitosis. It's as if the cell has received a signal, even though it hasn't, and cells proliferate out of control. When cells proliferate out of control, that's the hallmark of the disease we know as cancer. There's another important interface into the cyclin E dependent part of the cell cycle. The DNA damage checkpoint detects, among other things, double strand breaks, produces a signal, the details of which I can't tell you because I've forgotten. But at any rate, the signal makes its way somehow to a protein evocatively named P53 because it happens to be 53 kilodaltons. Well, P53 is a transcriptional regulator excuse me. Let's give it a little phosphorylated tail here. And this transcriptional regulator targets a number of things, but in particular, inhibitors of the cyclin-dependent kinases. One of which is a small protein called P21. If I'm representing the cyclin CDK complexes as sharks, P21 is basically a pseudo-substrate, a surfboard, let's say, such that this kinase encounters, instead of its normal substrates, it encounters this pseudo-substrate and gets jammed up with it, thus inhibiting the cyclin E CDK2 complex from going and targeting the DNA replication machinery that it would otherwise have to phosphorylate in order for cells to transit S phase. Better fix that before you try to replicate it, right? Finally, let's go back to the spindle assembly checkpoint that's operative during mitosis, in which the kinetochores monitor tension. This kinetochore pair here is attached to kinetochore fibers, it's under tension, it's quiet. Whereas this one here, let's say, is at least not under tension yet, and it is emitting some sort of signal broadcasting, wait for me, I'm not ready yet, don't proceed. So the inhibitor that it produces is a complex between a little protein called MAD2 and another small protein, CDC20. CDC20 is an essential component of the APC. If I draw the APC as this great big hexagon here, let's imagine that there's something that needs to fit into a critical slot, much like a key fits into the ignition on a car in the old days before key fobs, right? So let's imagine we've got a car key fitting into the APC. That's CDC20. And in effect, the signal produced at an unattached kinetochore is a complex between that's supposed to be the Toyota logo, sorry. A complex between MAD2 and CDC20 so that it cannot fit into and engage the APC. So that is the nature of the so-called SAC, or spindle assembly checkpoint. These are three key steps gating the transitions through the complete somatic cell cycle. They are not the sum total of them, and I want to mention in closing one of the most recent discoveries in cell cycle control, which has to do with the control of cell growth. A couple decades ago, someone prospecting on Easter Island for interesting new microbes got a soil sample that included bacterium Streptomyces hygroscopicus, which secretes a lot of stuff, including a potent antifungal that they named rapamycin. Rapa for Rapa Nui, and mycin because it inhibits fungal growth. It arrests fungi in G1, yeasts and so on. It also turned out to be clinically useful as an immunosuppressant, and the target of rapamycin turned out to be a protein kinase, now known as TOR, for target of rapamycin, initially discovered in fungi. When people found a mammalian one, they at first called it the mammalian homologue mTOR. This turned out to be a universal control. So now the M stands for mechanistic target of rapamycin. It turns out to be two of them. This kinase integrates signals about nutrient availability, energy balance, oxygen, etc., and sets a switch for growth versus autophagy. That is, if you have enough amino acids around, if things are fine, and you can build stuff from new resources, growth is good, at least as long as we wait for a signal to actually divide. And mTOR works in part by turning up ribosome function. On the other hand, if amino acid deficiencies are detected, mTOR turns down ribosome function in favor of lysosome function, the little bags of death water referred to in the thing explainer. One of their functions is to chop up proteins, even your own proteins, and convert them to amino acids that you can use for cell maintenance. Okay, so mTOR mediates this switch between growth versus autophagy. Autophagy means self-eating. And this is the last major piece discovered for eukaryotic cell cycle control, and still perhaps the least well understood.